Yeah, I'd like to present some results of my PhD thesis, where I used so-called finite-difference time domain simulations to explain effects that we observe in our microscopy measurements of brain tissue. So we use a microscopy technique called 3D polarized light imaging, or PLI in short, and here we place histological brain sections between a pair of cross-linear polarizers and a quarter wave retarder. And from the phase and the amplitude of the resulting signals, we extract the three-dimensional orientations of the nerve fibers, which are here encoded in different colors. Here you see how I simulated the PLI measurement for two simple fiber configurations. So I model the light source as a plane monochromatic wave with a certain wavelength and angle of incidence with circular polarization. The sample is represented by an artificial nerve fiber configuration, the other polarizer by a matrix, and then I compute the transmitted light intensities. The propagation of the light wave through the sample is simulated by so-called finite-difference time domain simulations. So here we have a spatial and temporal discretization, and then we approximate Maxwell's equations by so-called finite differences. So I cannot go into detail here, but you can come and see me at my poster later on. So the mesh size here needs to be much smaller than the wavelength. So this is computationally very intense, and I performed my simulations on the supercomputer UQueen at Research Center Jülich, consuming up to 19 million core hours per year just for my project. So now to give you some examples how I use this type of simulation to explain effects we observe in our measurements. So here we measured striatum fibers in the caudate putamen of a coronal mouse brain section, and we had a look at the transmittance, so the average transmitted light intensity. And here you see that the transmittance decreases with increasing inclination angle of the fibers, which were here determined by two photon fluorescence microscopy measurements. And I could explain this effect when I simulated fiber bundles with different inclination angles. So I had a look at the scattering of the light, so how the light is scattered when it passes through the sample. And you see that the scattering increases with increasing inclination angle of the fibers. And our imaging system has a very small numerical aperture. So the camera only detects a small portion of the light. And this leads to a decrease in the transmittance. So it's a bit hard to see, but here the solid curve belongs to this densely grown fiber bundle, and the dashed curve to this bundle with broad fiber orientation distribution, and this gives us a similar decrease as we also observe in our experiment. Then I also had a look at crossing fiber bundles, and you see that the scattering pattern shows the underlying crossing angle, and the transmittance does not depend on the crossing angle of the fibers, and as I said before, it's much larger than the transmittance for the steep fibers. This we can also see in the optic chiasm here, where we have two crossing fiber bundles. So the birefringent signal here cancel out, but the transmittance in the crossing and the parallel region of fibers is almost the same. And this we can now use to distinguish regions that all show low birefringent signal. So we identify the region with the maximum birefringence, because here we expect that we have a maximum of fibers, so a maximum of absorption. So when we now have a region with similar transmittance, we expect that this region contains in-plane crossing fibers, the region with lower transmittance to contain steep fibers, and a region with higher transmittance to contain less fibers. Then I also used this finite-difference time domain simulations to explain something that we call diattenuation. So here we simply pass linearly polarized light through the brain section, and we observe that in some regions, here marked in green, the transmitted light intensity becomes maximal when the light is polarized in the direction of the nerve fibers. And this effect I call D+. And in other regions, here in magenta, the light intensity becomes minimal. And this effect I call D-. And here you see now the diattenuation for a coronal and a sagittal section of a vervet monkey brain. And here the corpus callosum, for example, shows the D- effect in the coronal section, and here in the sagittal section it shows the D+ effect. So this effect actually depends on the inclination of the fibers. And we also observe when we embed our brain sections and then wait for a longer time, we see that more regions show the D+ effect. And this I could also explain with the simulations. So I used the simulation for the diattenuation caused by anisotropic scattering, and for the diattenuation caused by dichroism, I used an analytical model. And you see that the anisotropic scattering actually causes this D- effect for flat fibers and the D+ effect for steep fibers. And we also expect, with increasing time of embedding, that the scattering decreases, so we are left over with the D+ effect here, caused by the dichroism. And this is also the reason why I observe more green regions after longer embedding time. And I can also reproduce the inclination dependence. So when we have steep fibers, also a bit hard to see, they all show the D+ effect in green. Flat fibers show first the D- in magenta, and then after a while here the D+. And for intermediate fiber inclinations, we see both effects. So I could use these finite difference time domain simulations to model transmittance and diattenuation in brain tissue, and I'm sure 7 minutes are not enough to explain my whole PhD thesis, so if you're interested, you can come and see me at poster number 1 this afternoon in the poster session. And with this, I'd like to conclude my talk. Thank you very much for your attention, and my colleagues and supervisors for their support.